Welcome back to 1E9, the conference, and welcome to all viewers from the High A Festival for Science and Art in Bavaria who are also joining us now. Because what's happening now is a cooperation between High A, the Deutsches Museum, and 1E9, and we can welcome three very special guests to this very stage, and we will even hand over an exhibit that has written medical history to the Deutsches Museum. First, let me introduce to you our guests. Joining us remotely are Professor Özlem Türeci. She's the co-founder and chief medical officer of BioNTech. She's a physician, immunologist, and cancer researcher with translational and clinical experience. Great to have you here with us.
Thank you so much for the introduction.
Please welcome Professor Ugur Sahin, the co-founder and CEO of BioNTech. He's a physician and immunologist as well, and a leader in the development of novel approaches to fight cancer and infectious diseases. Great to have you up with us tonight. Thank you very much for that. And we are happy to have another pioneer of mRNA technology with us, and even in person in Munich. That's just amazing. Welcome to Dr. Katalin Karik, who as a biochemist specializes in RNA-mediated mechanisms and who joined BioNTech in 2030. Great to have you here.
Thank you.
Before we talk to you and hear a lot of amazing stuff, let's have a look at a greeting from the Bavarian State Minister for Science and the Arts, Bernd Siebler, who initiated the High A Festival.
Dear Dr. Türeci, dear Professor Sahin, dear Dr. Kariko, dear guests. Vaccines based on mRNA have highlighted the importance of science for our world. 2 years ago, only experts knew about the potential of this impressive technology. And now your vaccine is helping us to protect people in a fast and relatively easy way. It has been a true game changer. And this is just the beginning, as the full title of today's event makes clear. This kind of vaccine may also help us in our battle against cancer. Without basic research, none of this would have been possible. We need a scientific system that gives our scientists the necessary freedom for their research, even if the outcome is unsure. The German Research Association has supported your basic work on mRNA methods. Before and beyond that, you have been persistent. Even in difficult phases, nothing could stop you. On top of your excellent scientific ability and your foresight, you have also shown courage. As soon as the first infections with COVID-19 became known, you adapted your processes at BioNTech. You made every effort to find a vaccine, and you found a very good name for your program, Project Lightspeed. You have provided great services for science and society. We cannot thank you enough. And I really hope that we can soon convince everybody to get their shot. Good science communication is key for this mission. This is why I'm such a big fan of events like the one today. In Bavaria, we also want to play our part in the development of vaccines based on mRNA. There is excellent immune research all over Bavaria. In Würzburg, we have got the Helmholtz Institute for RNA-based infection research. We have also got a new strategic alliance with sites in Regensburg, Erlangen and Würzburg. This shows that Bavaria is the perfect place for today's outstanding exhibit. The bioreactor in which you have created the very first batch of vaccine is truly special. And the Deutsche Museum provides the ideal setting for it, especially if we bear in mind that its name translates to German Museum of Masterpieces of Science and Technology. Thank you for this honor. I wish you all a lively panel discussion and a wonderful evening here in Munich. Yours sincerely, Bernd Siebler.
Thanks, Minister Siebler. He already gave a teaser of what we are about to unveil here, but please forget it, so it's a surprise in about 1 hour. Now, it's been less than a year since the COVID-19 vaccine from BioNTech was approved in the US and a bit later in Europe too. And from the outside, it looks like a lot for you has changed. You've won many awards. You're asked interview guest for everyone. Your company is successful, but the road to get there was not always that easy. There were setbacks and there were doubts from others. So maybe what I want to know first, what was your motivation and your drive to invest so much time and energy in your research? And this goes to all three of you. Maybe you want to start.
Yeah, so science is like that, it's not like you do an experiment and you get the result and immediately you proceed. So that it is taking time to understand the system and perform experiments. If you have a improvement and you can see the product is getting better, you get more protein in case of RNA, then you are proceeding. So it's not like you keep doing something and just repeating and get no further. So that is important, and it is how science works.
What about you, Mr. Türeci? What drives you to go on?
My motivation was that this is the same for Ugur Sahin, that as cancer physicians, we saw the suffering from another pandemic, from cancer. And we wanted to translate our science into survival for cancer patients. And this was the motivation which drove us all those years to invest time and efforts in developing the mRNA technology and other immunotherapeutic approaches. So motivation was to help against suffering.
You said it was for both of you, Mr. Sahin. You want to add something anyway?
Yeah, I think one important piece in science is people think that science is about knowledge. But a lot of science is about imagination, and imagination is even more important than knowledge at the beginning, because we of course when we started, as Kati said, yeah, we things didn't work, yeah. So, but we imagined how we might be able to use mRNA, and we believed in mRNA could work. And the gap between what we imagined and what we see every day is the driver and the motivation to continue to improve, so that what we imagined could become real.
Thanks for that. Now let's quickly jump right into the amazing story of how you developed the vaccine in record time. One of the key moments was in early November 2020, when you two, Özlem Türeci and Ugur Sahin, were waiting for a call. Do you know which call I mean? And if so, why was it such an important call?
Yes, we know what call you mean. It was the call which was the moment of truth, namely the call which would inform us about the outcome of our phase 3 clinical trial, in which we compared together with our partner Pfizer our vaccine against a placebo. And until that moment, it was there was no way of knowing whether the vaccine would even work and prevent disease. And this call, which came on a Sunday evening, was one which was like a blessing, because we learned that our phase 3 trial was positive, and we did not only show efficacy, but showed tremendous efficacy of 95% against COVID-19 disease.
And what did you do when you heard that the vaccine actually worked? Did you celebrate wildly, or what did you do?
You know, it was a very these were very tough months for every one of us, for our team and us. So we were exhausted to celebrate too wildly. But there was some jumping up and down and some tea drinking and congratulating each other.
Oh, that sounds like a crazy party. Katalin Kariko, after decades of research in RNA and mRNA technology, how did you learn about this breakthrough, that it actually worked, and what were your thoughts then?
As just Özlem saying, it was Sunday, I remember, because November 8th is my daughter's birthday, and we celebrated with my husband, and then Ugur called, and it was like afternoon, because I was in the US, so 6 hours. And he asked me that whether is there anybody else in the room, and then he told me that it worked, and so I was kind of telling my husband that it worked. And I ate the chocolate-covered peanut, which is my favorite, and usually even gave my colleagues here in Germany, I bring them as in a movie theater box, and I told my husband that I will eat the whole thing. And anyway, so that's how I celebrated, and I was very happy.
So that was a bit more than tea. I promise everyone in the audience that we will get back to Project Lightspeed. That's how the development of the COVID-19 vaccine was called internally at BioNTech. But first, let's talk a bit science. Özlem Türeci, Ugur Sahin, one of your most important investors, said in an interview that he was particularly impressed by your special gift of explaining complex issues in a simple and understandable way. Maybe we can get a small example of that, since we are going to talk in more detail about mRNA technology. Maybe you could explain one more time in simple terms what mRNA and mRNA technology actually is. Ugur Sahin.
I leave that to Özlem because she can explain it even better.
Okay, I can take over here. Vaccines have the purpose of alerting the immune system against the enemy and preparing thus an immune response. And mRNA is the most ancient information technology. So what it delivers to the immune system of the vaccinee is basically the printing template for the enemy, something like a wanted poster. And it not only delivers this information, how the enemy looks like, but it also delivers the information that an attack is needed and that this is a danger signal. And mRNA is very versatile, which means it can be easily manufactured because it's a natural product. And the combination of these features makes it so favorable for the use as a vaccine.
But as we will learn later on, also it has even more potential to revolutionize medicine, not only for vaccines, but you were originally working on finding a cure against cancer. So what's so special about it that it has this wide field of potential use cases? Maybe, yeah, Ugur Sahin, yeah, maybe you take this one.
Yeah. I take one part and Kati takes the other part. So at the end of the day, as Özlem said, mRNA is a molecule which can be used to transfer information, to instruct cells to make proteins. And of course, since the protein could be very different one, we imagined that we could instruct the immune system to activate the immune system against cancer, so that this would be a therapeutic cancer approach. But we imagined also that we could deliver other type of immunotherapies, for example, antibodies attacking cancer cells. And actually, Kati, when she started research, had even another application in mind. But Kati could tell that even better.
Please do so.
So I was not interested in vaccine, not cancer vaccine or infectious disease vaccine. I want to deliver mRNA coding for protein, which actually is already in our body, but maybe not enough, not the right place, and which would accelerate healing and curing the person. And so I want to make this kind of therapeutic protein coding RNA, and that was why I was working on it, and of course not realizing for a while that this RNA is actually not feasible for that kind of application. But it was a later story.
And now, within the last few months, this knowledge about the potential of mRNA technology has become kind of common sense, at least in most parts of society, but it wasn't always like this. So maybe you can tell us, as a young researcher, when did you start researching RNA and mRNA? What were your thoughts? Did you think, okay, this looks interesting, I'll do research for a couple of years and then I'll have have a cure, or how did it all start?
I mean, I started working with RNA in Hungary, but those were just short RNA which had antiviral effect. It was not mRNA. In '89, in University of Pennsylvania, I started to work with mRNA and synthesize it. And the colleagues usually were actually not very critical. Actually, when I said I'm working with RNA, they kind of said that poor Kati, you know, they felt sorry. Because when they had to work with RNA, it's always degraded, and they thought that it is too labile, and so they felt sorry for me. But I in my hands, it did not degrade it, because in the laboratory I did not work with an enzyme which would degrade and didn't contaminate the room. So I was working in it, and I could detect improvement, more protein was made, and tried to figure out. Actually, I did basic research, but I always thought about that, it would be used for something. But the protein made from the RNA in a cell, later in animals, it was too small amount. And then for therapeutic purposes, so try to see that how it could be improved, or whether find a kind of a disease where a small amount is sufficient.
You already said that, you know, some other researchers were saying, oh, poor Kati. In Hungary, I think you even lost your job as a researcher. You emigrated to the US, to the University of Pennsylvania. But even then, some grant applications and papers were rejected, if I got it right. But I think it's great because at least that's what I get from the stories. Now I'm talking to you personally, that you weren't demotivated, because as the story goes, you said to your later collaborator, Drew Weissman, when you met him by chance at the copy machine, let's clarify if that's true, and he told you that he tries to develop a HIV vaccine that said, I can do that with mRNA. I don't know if that really happened. Maybe you can clarify.
The machine was good, and maybe I did an outlandish statement. I just recently heard one colleague who was studying surgery, and I told that by that time it will be obsolete because RNA will be replace everything. So maybe I did an outlandish statement, but really I just offered to Drew that I can make RNA, and that's what I was bragging about, that I can make RNA, and so that's how it started.
Yeah, and now you get all the prizes, as well as you, Özlem Türeci and Ugur Sahin. You've also been researching mRNA technology for more than 2 decades, and initially you focused on developing individualized treatments for cancer patients. Why did you focus on cancer treatment in particular, and why did you choose mRNA back then? Because back then it wasn't like the mainstream thing to do.
So cancer, why we focused on cancer, the answer is very easy, because we were cancer physicians, and we could we would experience every day that what we could offer our patients, which was at that time in the early 1990s, chemotherapy, radiotherapy, or surgery, that this was not much what was in the armamentarium of oncology in those times. And on the other hand, at the same time we were working in the lab and were researchers, we experienced that there is so much science, and in particular immunology, could offer. So one thing which we believed in was that if we want to develop an ideal cancer treatment, we need to harness the immune system, because in principle the immune system can be very potent. It has the adaptability of a predator, and at the same time it can act very precisely. And in order to instruct the immune system to do all this and fight against cancer, you need a cancer vaccine. So it was very clear we would need to develop vaccines, and a vaccine needs to be very potent, because at the time cancer is diagnosed, it has already invaded the body. There are billions of cells, and they are heterogeneous. And that again means that we need something which is individualized, because every patient's tumor is unique, and the ideal situation would be to have a cancer vaccine which is tailored and on demand produced according to that individual patient's tumor and genetic makeup. And we tried different platforms and understood very quickly that mRNA would have the potential to address all those needs for a cancer treatment. That was the beginning. And however, it took a long journey because we needed to optimize mRNA on different levels in order to actualize the full potential as a cancer treatment.
It's a pretty complicated stuff for people who don't work in science. For people who are not working inside it, it always seems incredibly long, how long medical research takes, especially if you compare it to the world of digital technology, you know, where companies like Google or Facebook just pop up. So maybe you could still take us on this journey a bit that you just mentioned. What was the problem or the challenge with mRNA that could not be solved, or maybe not perfectly solved, for so long, and how did the three of you finally solve it? Maybe Ugur Sahin, you want to start?
Yeah. So there is not only one problem. It is at the end of the day, if you want to accomplish something like making mRNA work, it is. You can compare that with the idea for building a first time a plane or building a rocket, and you need to solve a number of technical challenges. So one challenge is to stimulate the immune system and understand what is really relevant to get strong immune responses. So what we learned from immunology is that a vaccine needs to be delivered to specialized cells, so-called dendritic cells, a delivery. So the delivery of the mRNA to the right cells is a problem. The second thing is that the immune system needs to be activated to not only to get the information, but be able to strongly mount an immune response. So we had to solve that. And the third aspect is that the message, the information must be appropriate, so that even delivering low doses could help to get strong immune responses. So it was the potency. Yeah. So that were challenges on our side, yeah, that needed to be solved. And as Kati said, we start to do experiments. We have hypotheses, we evaluate, and in 80–90% of these cases, we find out that these hypotheses are wrong. Yeah, and sometimes we, by chance, make findings. Yeah. So, for example, we made by chance the finding that something we wrongly cloned, turned out to work particularly well, by the discovery that instead of one element that we wanted to clone, two elements were in the mRNA. So we came up with the idea, can we combine elements? So these are the way how to deal with that, having a challenge, having different challenges, and trying to solve the challenge by modifying different aspects of mRNA. And I stop here, and Kati could take and the complementary part.
Yes. So one problem was, of course, we mentioned that it is very labile in when you inject in the body, and the other is even when it's cell you inject, it was degrading quickly, and so the encoded protein we can see short period of time. So when with Ugur, we visited one big pharma company, and they showed that every day it was 10 times less protein was made, and they told us that if you can change that and every day it will be the same, come back. You remember, Ugur, that? And so that was one thing, and the other is for investor who wanted to invest money. Then they criticized that there is no shelf life. So they said that if you can put something there and for 2 years you can take out aliquots. and then you can make trial and other things. So that was also not solved. How you formulate and keep it there. And another thing that most of the other scientists were trying to use the mRNA for a problem, which the gene therapy problem, and they wanted to more longer period of time to get the translation. Today we know that how good that, the spike protein is just made for a short period of time. Nobody wants to do rest of their life. So it is good that it degrades. It's good that it is just a short period of time. But at that time, early on, people tried to use for gene therapy purposes. And that's also why people thought that, it's just too short of the effect. So many level was the problem.
It seems like it was a complex thing. We are glad that you achieved it to solve it. Now, as you said, you were talking to investors, you were visiting other companies, because maybe the solution or the solutions that you found would be of less use to us in the current pandemic if you had, just published, as you did, scientific papers and not built a company like BioNTech. When you two co-founded the company, and you two co-founded the company in 2008, you joined it in 2013, and this step from academia into a company, especially in Germany and Europe, that is too rarely ventured. That's what many critics say. Why did you decide to do it anyway? What was the reason to say, okay, we step out of academia, we still do research, but we do it within a company?
Yes, it was in principle the strong determination to translate our science. As I already pointed out, we are physicians and scientists, and the motivation is to bring indeed the science from the lab benches to the patient's bedside and help against suffering. And we became founders and entrepreneurs out of desperation, because we learned that there was no other way to ensure exactly this translation.
And what was the please.
So the limiting factor is, if you really want to develop drugs to be tested in patients, you need a huge amount of investment. So to prepare a novel approach for clinical testing, we had to set up the manufacturing of mRNA. Of course, in the lab you can relatively quickly do for experiments with laboratory quality based of mRNA. But if you want to have the same mRNA for clinical application, you have to invest tens of millions of euro, just to build a manufacturing to ensure that the mRNA has the right quality, before you are able to do a clinical trial. So it was very clear that this financing of this project would not be possible in the academic setting. What is also important to understand is that at a certain limit, if you, for example, have a solution, an mRNA, which qualifies for a certain application, then it is okay from the academic setting to have 80% of the problem solved. But the last 20%, solving the last 20%, really means additional 10 times investment, because the quality considerations and the requirements for developing drugs are so extremely high. So that was the reason that we decided to go into a company, to start a company, and we were happy and lucky that we got the funding from investors. Who were not classical investors, but investors who really also wanted to help us to develop new treatments, innovative treatments, and that allowed us in the first 6 years to build the technology, to build GMP manufacturing, and to enable to start our first clinical trials in 2012.
I just want to mention, so I myself also in 2006 with my colleague, we established a company because we wanted to develop further, but it was just a start and we couldn't get funding. And the main reason actually I came to BioNTech because, and people question that, you know, the wrong direction. Usually people go to the US if they, and I came here because I learned that at BioNTech already has clinical trial, the mRNA, and because I wanted to develop the messenger RNA, which contains this nucleoside modification to clinic, I thought that it would be the best place to come. And because already there is manufacturing, there is already clinical trial, and I was just so impressed with the kind of thing that I learned that here in the company, we have to make a product that is important, and everybody works together. And I also realized that how much work is to make a company, and I watched Ugur and Özlem that how they did, and there's a lot of work.
So, as you said, that's a very unusual stories that you know, researchers go from the US to Germany to join a company. Now, maybe you already mentioned some of these, but maybe you can talk about the factors that contributed to your success, because I guess we would all be happy if we see more companies like BioNTech. So, what were like the success factors that you say, okay, that's the foundation of where we are now?
I start, then. So I think one success factor is really focusing on the problem. So BioNTech is for sure not the usual biotech, which comes with the idea of bringing a product as quickly as possible into clinical testing and hoping it will work. So we really invested many years before we started our first clinical trial, and we were always scientifically, from the very beginning, scientifically driven. That was also the reason why for example, we were interested that Katie joins us. So we had some industry expert, but we really recruited scientists, from academic institutions, from Max Planck institutions, from universities. Katie came to us, and we realized that we have like-minded people in our company who in a scientifically driven and fully dedicated, committed manner, tried to solve the basic challenges to enable that we get a highly active mRNA. So that is one success factor, really focusing on science.
Any other factors that we might learn from?
Yes, Ugur, these were two factors: science and the focus on science and being led by science, and the other one you named, which I would like to also echo, is people. And this starts from our investors. We chose the right investors who were committed to the mission of the company. We had the right team with people who were scientifically focused, and we also chose the right business model, namely not in a fast-focused, lean way, just focusing on one product and developing it as fast as possible, but having a technology platform or several technology platforms in which we iteratively invested for improvement.
Thanks for this advice. Now, as I promised, we'll come back to Project Lightspeed now. I know you've had to tell this story many times before, but it's historic and it's just a great story. So I'm sure many here would like to hear it too, at least the beginning. Ugur Sahin, in January 2020, most of us still, me included, thought COVID-19 had nothing to do with us in Germany. In Europe, the outbreak will certainly be contained in China soon. And yet you already came up with the idea that your company, BioNTech, which was actually focusing on things like cancer treatments, could and should now produce a vaccine. Why?
Yeah, so I was also not concerned at the mid-January about this outbreak in China, but in end of January I read a Lancet paper which came out the same day, yeah, and in the Lancet paper this outbreak was described in a extremely clear fashion, and the outbreak had all the criteria for becoming a global pandemic. And a new virus, highly infective, in a Chinese megacity with high travels. And one of the key aspects that were evident from this paper, that this virus induces asymptomatic infections, which make it impossible to control and keep the pandemic in the localization. And we already knew on the same day that we will encounter a global pandemic in the coming weeks. And the question was, can we develop a vaccine? And since we had a powerful vaccine technology allowing us to induce strong immune responses, and since we had built the ability to really quickly come up with a vaccine because of this personalized immunotherapy approach, so we had implemented in the company the ability to develop cancer vaccines within a few weeks. Yeah, and we said if we can develop cancer vaccine in a few weeks, it should be also possible to make mRNA vaccines against this new virus in a few weeks. And so that was the belief that we can do something against this pandemic, and it was an obligation to start a project because we knew that we could be among the first companies and groups worldwide coming up with a potentially effective vaccine.
Now, we all know now that it worked out, but Özlem Türeci, what was your first reaction when your husband told you that he was proposing to even risk the future of your company to develop a vaccine against a disease that many thought was not our problem here?
Well, he had science on his side. So when he shared his concerns, he basically shared a mathematical model which was very convincing and which basically said that we are already in the midst of a pandemic. And it was even scarier because he offered two scenarios. One scenario of a slow ramp up of the pandemic, which actually was the scenario which then at the end became real. So the one through which all of us had to go. And the other scenario was a very fast evolving pandemic, which would not give mankind any time to prevent that we all would be decimated. So therefore there was not much work he needed to do to convince me and also convince others, our team at the company.
How about you, Katalin Kariko? When did you hear about this plan and what were your thoughts?
Yeah, I mean, I heard about what happening in China, and I was, not thinking also that it will be here, and Ugur had this vision that foreseen that maybe it will happen here and coming, and it will be a pandemic. So I was not such a visionary to predict that, and so I was not concerned. But luckily he was, and Özlem, and so that's why it happened that started the project.
Now the rest is kind of history. You and your team developed the vaccine in record time. You ensured that it could be produced and administered in billions of doses. For those who want to know more about this history, I highly recommend the book you two wrote with Joe Miller. It's called Project Lightspeed. But I want to talk with you about the future as well, because now we all know about mRNA technology, so let's talk about what's possible with it and what you are planning. But maybe Katrin Kariko, you answer a question first, because in interviews that I read, you've said that at the beginning of your career you had the idea that at some point we will have mRNA in our refrigerator to treat, for example, blisters on our feet. How do you envision our future with mRNA now?
Yeah. Actually, my daughter is a rower and she had all the blisters in her hands, and I was thinking about that, what kind of mRNA could accelerate healing, and I found that collagen 7 protein can accelerate. And it is also interesting because, you know, we are talking about here, not the mRNA is really the final product which will heal, but the protein is coded. And the protein, that protein actually half-life is very long, so even the RNA is gone, the protein can be there for months. So I was thinking about this accelerate the healing, and other things were also like work with colleagues that working on pain killing, but it was a natural way. And so I thought that most of us would need, you know, some treatment acute disease, which aches and pains, and therefore I was not thinking about problem immunogenicity, because I thought that we just have to have the same protein which already we have in our body. And so turned out, of course, that the RNA we are making, the basic nucleotides, those are inflammatory. So I spent so much time to make it with non-inflammatory with my colleague at Penn, and then so this kind of RNA, I was thinking this kind of simple diseases, which is really, I learned from Ugur actually that it is very difficult to run a clinical trial for things that heal on their own, because it is difficult to measure something. And I learned so much that I get good ideas, and then I realized that it is not that good, because it is difficult to run a trial, difficult to implement and measure something that it is effective. So, but there are a lot of things that whenever I listen to a lecture, somebody talks about a disease, I think that, oh, it will be good to treat with RNA.
So some examples?
Yeah, I just said, you know, pain. Actually, our first grant we submitted was for treatment for anemia. So that, again, a protein, the erythropoietin coding mRNA, it was an animal model, and we demonstrated that the hematocrit increased, more red blood cells was produced. And so it is, again, just transiently applying the mRNA coding for the protein, and then you have biological effect, and then you just stop. It is more like a conventional medicine. You treat, you use for once or twice, and then fix the problem.
Özlem Türeci, Ugur Sahin, when you co-founded BioNTech, as we said, to develop individualized cancer therapies, now BioNTech is highly successful with vaccines, as we all heard. You could say, okay, let's just make this kind of vaccine forever, but I guess that's not the case. So will you still continue to pursue this goal of the cancer treatments that you started with, despite the success of this COVID-19 vaccine?
Definitely, we will do that. That was and still is our original vision. We were a bit distracted now with the pandemic, and had to help to solve it in order to now go back to our original mission, which we never sort of paused. So cancer vaccines and other types of cancer immunotherapies based on mRNA will continue to be very close to our hearts, and we will develop them. And beyond that, there are other areas in which we believe that our mRNA technology, now in its further refined form with the experience of the Lightspeed project, can help. These are, for example, other infectious diseases, malaria, tuberculosis, and HIV, all pathogens which are with mankind for much longer than the coronavirus, all pathogens who have developed mechanisms to escape our immune system actively. These are also areas in which we want to use mRNA, and there's also many indications beyond that. One indication we are particularly interested in are autoimmune diseases, such as, for example, multiple sclerosis, because as we already pointed out, you can use mRNA to tell the immune system what to attack, but you can also use it to tell the immune system what not to attack, namely those self proteins which are attacked in the case of autoimmune diseases.
Now we all learned that it can be pretty fast to develop a new vaccine, a new treatment. Now, maybe you could give us a timeline on the projects that you will say, when will we see results, when do you expect treatments and new vaccines here?
So our cancer vaccines are already in phase 2 of 3 phases of clinical development through which a drug which is developed has to go. And these phase 2 trials are very important because in those one has to compare against the standard treatment for that respective disease or cancer indication. And as soon as we have data from those trials, we will know how long it takes to get to an approval. That could be within the next couple of years, the next 3 to 4 to 5 years.
Ugur Sahin, no, it goes to all of you. Now, you get all this recognition now for your work. You won those prizes. Everyone, you know, many people are very thankful for what you did, you know, that you kind of saved many of us from a horrible pandemic. From this recognition as a scientist, and I think it's great you still stayed scientists and researchers, what was most important to you personally? Ugur Sahin, maybe you start.
Yeah, personally, to be honest, we feel blessed that as a scientist we could make a difference. Yeah, so because all of us started with the idea to develop something which could help people. Yeah, and the greatest prize we achieved is that we were able to make a difference. Yeah. And everything else is wonderful, but nothing matches this feeling that you know that you contribute to something. And of course, it is not only the 3 of us. It was a huge scientific community. If you really ask for each of the details, you most likely end up with several thousand scientists that have directly or indirectly contributed to the success, yeah, and this progress of the mRNA field and the making of the mRNA, of the manufacturing of the mRNA. But we feel really blessed, and that during our life we were able to make a difference, and that's the greatest award that a scientist can get.
Maybe you remember an incident where someone approached you or where someone thanked you. Was there moments like this when you really could feel it personally, that you made this impact? Any of you?
Yes, we got, for example, a picture of a grandfather with his
Son.
With son and the kids, yeah, and they wrote us they were more than almost 1 year apart and could not join, and they were able after the vaccination to rejoin, and it was somehow a great family picture, showing the reunion of this family. And when seeing this picture, I realized that there are many personal stories of people who got vaccinated, yeah, and who have this life-changing event, yeah, to be able to go back again to a almost normal life.
Yeah, actually, I also got an email from the Meadowbrook, New York State elderly home, and then they reported me that 1 week after I received the vaccine, which was televised, 1 week later, all of the residents received, and there were 200 residents there. And but 1 week later, they already found that some people get infected, and before that, it was when in an elderly home somebody get infected and others get. At the end, you know, a lot of people died, and they reported me that nobody died, and so were they so happy, and they made a Katalin Karikó Appreciation Day in September, and everybody had a T-shirt with my picture, and they sent me that photo. And of course, you know, similarly to Ugur, you know, and Özlem, that we know that so many people contributed, but some people just happy to point out somebody and say that, oh, I thank to them. But we know, all of us, that a lot of people. And myself, I feel just, you know, happy to know that I am one of them. And when I did the research also, I never was important that it is my name or me. It was, I was happy that I was part of the project. And many times we mentioned that, you know, the patient never asked who developed the drug. They just asked the doctor, do you have something for my disease? But now that we are here, and now people start to know that, oh, there are scientists, and it is also a good thing that we are here today, for example, that people realize, yeah, these scientists, our lives depends on, and maybe, you know, they can name at least 1 or 2 scientists who are still alive today, because when you ask a person, name a scientist, you know, maybe say Einstein, and that's it.
Yeah, we could walk over to the museum and see a lot of famous but dead scientists. Also living ones, absolutely. Ugur Sahin, Katalin, you shared your personal stories, and it's just so great to hear personal stories. So maybe Özlem Türeci, you have one for us too.
Oh, there are so many. I would really not know which one to pick. We get so many emails. We got, for example, a video clip of hospital staff who danced for us, and one could really feel how relieved they were, because after vaccination it was easier for them to take care of their patients. We get pictures drawn by kids who thank us, and the example Ugur just noted with grandparents seeing their grandchildren, even newborn grandchildren, where only after months they could meet this new member of the family. So there are so many stories.
That's great to hear all of them, even especially in times like this when it's not yet over, but we still stay optimistic. Now, as I promised, and as the minister already teased, we will hand over a historic object to the Deutsches Museum, and we will get it on stage now, and we will also get someone you already heard a second ago, Professor Wolfgang Heckel. Please come on stage, join us on stage, the Director General of the Deutsches Museum. We stand up, and now those who will unveil the object that is about to become a part of the exhibition of the Deutsches Museum. And there,
here we go.
It is.
Yeah, you may clap your hands. Thank you so much, Katalin Karikó, Özlem, Ugur. We are really proud to receive this masterpiece of science and technology. The story of the Deutsches Museum is, the duty of the Deutsches Museum is to tell the story of mankind alongside with these kind of masterpieces of science and technology. And this is a true masterpiece of science and technology because it has proven. And I remember when Özlem and Ugur were a couple of weeks ago in my office and we discussed about my research, the origin of life, that you are true scientists and you made a difference with such scientific achievement in such a light speed time. And you are now part of Benz motor wagon. You're part of Röntgen's first X-ray apparatus. You're part of the first quantum chip, the Sycamore chip from Google. And this is all based, I guess, and the story is really that we can tell our 1.5 million visitors a year that science, technology makes a difference and it can save lives. I was so impressed when you, Özlem, you told me that you have at that time, a couple of weeks ago, I think I remember, produced 43 kilograms of mRNA and it saved 1 billion people's life. Wow, what can be greater than that? Yeah, you really deserve to be part of the story of the masterpieces and the masters of the technology and science in the Deutsches Museum. Thank you so much, Katalin, Ugur, Özlem.
And thank you from my side to all four of you, but if I may quickly jump in now, we see that it's a masterpiece of science and technology, but maybe we can learn what it actually is, because it looks kind of weird.
It's a kind of art.
Yeah.
Science and art and technology have something in common.
So maybe you could...
Please, Katalin.
So this is a bioreactor. This is how the RNA was made inside this, in this container, and all of these tubings is how the components are added to it. Ugur, you want to say something
about this special bioreactor?
Yeah, absolutely. This is the first step of manufacturing of mRNA, bringing an enzyme and DNA template and all the components which are required. And it takes a few hours and that the DNA is copied into mRNA, and every DNA is copied into multiple mRNAs. And our team was in the last 10 years. Our teams worked so hard when we started to do our research. It was just 250 microgram of mRNA that we could get from such a reaction. And now, in the meantime, we are able to get from one reaction more than 250 gram or even more, which is a scale up of 1 million. In the production, and that was also needed at the end of the day to ensure that so many people could get their vaccines.
And maybe we have to point it out because this is actually the bioreactor that the first doses of the vaccine that we all got were made, right?
Exactly. Yeah.
Exactly.
Yeah, and I had a bit the impression, Wolfgang, that you were disappointed that we announced the bioreactor, and it had this underwhelming size, right? You expected to get something on a truck transported into the museum, but this actually shows how potent the mRNA is. This is also why we only need 30 micrograms per dose of the vaccine. And this is also the reason why we were able to deliver so many doses in such a short time, this underwhelming size.
Özlem, you are absolutely right. 5 hours ago, I got a shot BioNTech here in my arm, and I didn't feel it. It was so small quantity. But museum pedagogics is about to explain why this item, this object, leads us to new horizons. And it is important to know that you just copied nature, Katalin. You copied nature, because in nature, small amounts of messenger RNA and not DNA, because DNA has to be safe. Yeah, it's copied into messenger RNA, and then it goes to the ribosome, and then the ribosome catches the transfer RNA and produces the protein and the spike protein. This is the story we have to tell here to our people, especially also to the young people who would want to follow you in your life career, because we need more of you. People, scientists who become entrepreneurs, who become people who help other people. 1 billion people, maybe now 2 billion people have got your vaccine. And this is fantastic. I can't say better examples as masterpieces of science and technology for the Deutsches Museum. I'm so thankful for us, for the Deutsches Museum, for all the people who will visit and know and get known to the story of you people.
Yeah, absolutely. Thank you so much, Katalin Kariko, Özlem Türeci, Ugur Sahin, Professor Wolfgang Heckel. I was never more excited before an interview. I hope it went well. Sorry if it didn't. I was very excited because
these are lovely guys.
Yes, I know.
These are scientists, and therefore they are good guys.
Scientists who made history and who really made a difference. That was an honor for me to do this. Thank you so much. Thank you for having us. And you guys out there, make sure to visit the Deutsches Museum in a pretty short time to see this object. Can you already tell us when we will see
next year, beginning of May.
Beginning of May, so come here in the meantime and then come back in May to visit Deutsches Museum. Thank you so much for the session. This was amazing. Good luck for your future work, and thank you so much. Yeah, this was the big mRNA session with a historic object that we delivered to the Deutsches Museum. Thank you for that. Bye to our visitors from the High A Festival for Forschung und Kunst. And for the 1E9 conference, I head over to Daria.
Thank you.
Thank you so much.
Yeah, wow. I'm still very excited. Speechless that we had the chance to actually welcome this amazing scientist here on stage. My co-founder, Herb, is going to join me. And what can I say? It's a wrap, guys. Wow. Let's give a big round of applause for our crew, for our partners, for our sponsors, for our speakers. We'll start with that.
If Herb doesn't want, I join you.
No, he's coming, he's coming.
Yeah,
so
I'm still kind of overwhelmed. So
yeah,
you
I am overwhelmed.
Sorry.
You see, it's all real. It's live, so we are overwhelmed here. But I have to say, remember when we sat together and we said, look, for this conference, let's reach for the stars. We are in the formal planetarium of Deutsches Museum, and here we go. We managed that, guys. I am just speechless. What do you think? What were your highlights of this 3 days?
Shall I start, or
Yes.
who wants? Okay, I may.
We need to come down.
Okay, so I was in the audience, so I'm a bit more refreshed. So let's just recap a little bit and reflect. So I mean, we closed this with Professor Heckel. And I think one major highlight also for me was the appearance of Mr. Heckel alongside with Vitalik Buterin, one of the co-founders and figureheads of Ethereum on the first day. So definitely that's something to look up again at 1E9 or maybe even the Deutsche Museum YouTube channel sometime soon. So that was definitely my highlight for the first day. Second day, I think Professor Achleitner was amazing with her very sharp observations on the current state of entrepreneurship, science, transformations, and so on. And I really enjoyed at the end Professor Enrique Solano with his, and I still memorize it. So he was commenting on this thing between science and art, and he said something like, so science and art are the two sides of a same coin, and the name of this coin is sensorial experience. So if you want to know more what this means, look it up at on our YouTube channel, I guess, at some time soon. And well, today I think in that respect as well, something that Ugur just said today that really kept now in my mind is that it's not so much about knowledge, yeah, in science, but imagination. And so that's also a good thing to say here in the planetary, where we look out to the stars and imagine what could be done. And yeah, so I think these three highlights fit well together. And so that's my recap.
Maybe I can continue. I think what is so amazing every time we have the conference is that we try to make science and technology tangible and not that abstract. And yesterday we actually have 2 startups, which are great examples for that, of course. So we had Juicy Marbles, who are the first ones in the world who created a plant-based high premium, high-end steak, which is also, you know, we tasted it, all of us together. And then we had Coca company, a Munich-based biotech startup, who have created a substitute for a coca bean, which is very CO2 intense kind of to grow and then to produce in terms of the chocolate. And that we actually also could taste yesterday. So I think technology doesn't have to be, you know, in science doesn't have to be someone in the stars. It's actually also real, and I really enjoyed that.
Yeah, I enjoyed all of those 3 days. The best thing about this job is to meet such fascinating people and hear them tell their story and you know, get this inspiration. That's really, it was a lot of work, and we had to cancel parts of it last minute because of the pandemic, which was very frustrating. But after these 3 days, it doesn't matter because it was great, and we met such great people and had the chance to hear their stories, to learn what they learned in their life, and that was really amazing. I can't say more at this point, but because it was, yeah, there was a thing. So I guess Wonders, thanks for all out you there watching us, joining us remotely instead of coming here. We really appreciate your many questions, your comments, and the love you gave us on social media. That's awesome. Thanks for all the people who worked on this. Thanks to the 1E9 team, the team of Safe Agency who supported us great over the last month, the team of Drop-In. Thanks to Dominik from Lichtgestalten for this great audiovisual experience in the couple, and thanks to all other people who supported it. Thanks especially to Deutsches Museum, Professor Wolfgang Heckler, of course, who made a great show with us. Again, to Simon Glocklehöfer, who supported us over the last month, and to all the other guys, perfect guys from Deutsches Museum. And thanks to our partners, Huawei, Airbus, Intel Ignite, We2 Ventures, XR Hub Bavaria, and the BMW Foundation and Respond Accelerator. Thank you. Without you, this event would not have been possible. And yeah, stay tuned for
the next year. We will be back.
And we will be back in summer without a pandemic.
Fingers crossed.